ايضا هي من البكتيريا اللي تتبع للعائله المعويه Bacteriology. Shigella, highly infectious bacteria, is one of the most infectious bacteria. An ingestion of as few as 100 to 200 organism will cause disease. يعني هنا الانفكتيف دوز لهذه البكتيريا تكون قليله جدا، ممكن 100 الى 100 organism انه يسبب الاصابه. Most individuals are infected with Shigella when they ingest food or water contaminated with human fecal material. Shigella can survive up to 30 days in milk, eggs, cheese, or shrimps. The disease is fecal or route transmission. According to the taxonomy, the genus Shigella belongs to the Enterobacteriaceae and has 4 important medical species, including Shigella dysentery, which is the most important and most serious form of bacillus dysentery cause. Shigella vaccinary, that causes shigellosis in underdeveloped countries. Shigella soni causes shigellosis in developed countries, and Shigella boydii vice versa. Morphology and physiology. This bacteria is small gram-negative, facultative anaerobe, coliform bacillus, catalase positive, oxidase negative, non-motile, no H antigen, possess capsule or K antigen and O antigen, non-lactose fermenting. This bacteria are bile salt resistant, that is for selective media for ferment and fermented glucose. يعني هاي من الصفات المهمة إنه البكتيريا مقاومة للبايل سولت، لذلك البايل سولت يضاف إلى الأوساط الزرعية لكي تصبح selective media لهذه البكتيريا، كون البايل سولت يطبق مجموعة أخرى من أنواع البكتيريا. Also, this bacteria is able to reduce nitrates to nitrates and nitrogen. Classification also can be made on the basis of mannitol fermentation. This bacteria can be divided into non-mannitol fermenters, Shigella dysentery, and mannitol fermenters, including other types or species of bacteria. Morphology and staining: short rods, non-encapsulated, non-motile, non-spore-forming, and gram-negative bacteria. This figure shows how can you diagnose or differentiate between the members of the genus Shigella. Can be used in non-lactose fermenting or non-motile organism, can be divided into mannitol positive and mannitol negative. Mannitol negative Shigella dysentery, while mannitol positive including other 3 species. Then we make lactose fermenting. The Shigella soni is late lactose fermenter, while the other is non-lactose fermenter. Then make indole test. By indole test, when bacteria indole positive, this means the bacteria, the causative agent of disease is Shigella flexneri, while negative is Shigella boydii. Cultural characteristics: all members of Shigella are facultative and anaerobe. Grow readily in cultured media, pH 6.4 to 7.8, at 10 to 40 degrees, with optimum of 37 degrees. After 24 hours in incubation, Shigella colonies reach a diameter of about 2 millimeters and are circular, convex, colorless, but moderately translucent, with small surface and entire edge. Growth on selective media: this and XLD media, they appear pinched or reddish colonies, while in hectoenteric agar, they give green to blue-green colonies. طبعا كما تكلمنا بالمحاضرة السابقة، XLD media تستخدم لعزل بكتيريا السالمونيلا والشيجيلا. And if the bacteria that presents salmonella, they appear as a black colony, while Shigella pinch to reddish colony on XLD. Habitat and transmission: Shigella species are found only in the human intestinal tract, carries of pathogenic strands can excrete the organism up to 2 weeks after infection and occasionally for longer periods. Also, Shigella can be killed by drying and transmitted by fecal oral routes. شباب اكو سؤال؟
واضح دكتور.
واضحة دكتور.
واضح ان شاء الله. جيد.
واضح.
Now, pathogenic determinants or virulence factor of this bacteria that help in the initiation of infection, including O antigen. O antigen presence in all gram-negative bacteria, but differ in their side chain or sugar side chain, which can be determined the ability of bacteria to cause disease. Ability O antigen help in the ability of bacteria to survive. The passage of the host defense and maybe due to antigen. Invasiveness, virulent, Shigella penetrating mucosa, and epithelial cells of the colon in an uneven manner. Intracellular multiplication lead to invasion of adjacent cells, inflammation, and cell death. This also lead to necrosis and appearance of blood in the feces. Cell death is probably due to the cytotoxic properties of Shigella toxin that interfere with the protein synthesis, cellular death, and resulting phagocytosis response by the host account for the bloody discharge of mucus and pus and shallow ulcer characteristics of the disease. Shigella toxin is the unique between the bacterial toxin because of this toxin has three types of action: neurotoxic, cytotoxic, and enterotoxic. Most toxin has only neurotoxic or cytotoxic or enterotoxic activity, but this toxin has three types of action. Pathogenesis, including penetrate through the mucosal surface of colon, colonic mucosa, and invade and multiply in the colonic epithelium, but do not typically invade beyond the epithelium into the lamina propria. Thin layer of fibrous connective tissue immediately beneath the surface epithelium of mucous membrane, eventually attached to and invade into M cells and fire parts of lymphoid tissue and lymphatic system of small intestine. Pathogenesis and immunity. Exotoxin, Shigella toxin is neurotoxic, cytotoxic, and enterotoxic, encoded by chromosomal gene. This means this toxin is constant in bacteria and housekeeping gene for this bacteria because of the presence on chromosome. Enterotoxic effect also Shigella toxin adhere to the small intestinal receptors, block absorption of electrolyte, glucose, and amino acids from the intestinal lumen and lead to diarrhea. Cytotoxic effect: B subunit of Shigella toxin bind to host cell glycolipids in large intestine and activate the 60S ribosomal subunit, inhibit protein synthesis, causing cell death and microvascular damage to the intestine, and hemorrhage blood and fecal leukocyte can be seen in large number in the feces specimen. Neurotoxic effects, including fever, abdominal cramp, are considered signs of neurotoxicity. Clinical syndrome: shigellosis range from asymptomatic infection to severe bacillary dysentery. Two-stage of disease: watery diarrhea changing to the dysentery with frequent small stools with blood and mucus. Tetanus, cramp, fever also presents. Early stage watery diarrhea attributed to the enterotoxic activity of Shigella toxin. Fever attributed to neurotoxic activity of the toxin. Clinical syndromes process involve ingestion, non-invasive colonization and cell multiplication, production of enterotoxin by the pathogenic bacteria in the small intestine. Second stage of the syndrome, including adherence to the tissue, invasion of large intestine, typical symptoms of dysentery, and cytotoxic activity of Shigella toxin, increased severity of disease and lead to cell death. Clinical features including fever, bloody diarrhea, abdominal cramps, symptoms, mucus, pus, convulsion, mild infection, lead to watery stool, bacteremia rare, and hemolytic uremic syndrome may be developed in advanced or untreated cases. Shigellosis is a major cause of diarrheal disease in developing nations, major cause of bacillary dysentery, severe second stage form of shigellosis, leading cause of infant diarrhea and mortality death in the developing countries. Also, Shigella occur naturally in higher primates, spread from human to human through the fecal oral route, less frequently transmission by ingestion of contaminated food or water. Outbreaks usually occur in close communities, and secondary transmission occur frequently. Now, how can you diagnose Shigella infection? This can be diagnosed by culture. Culture is the gold standard method for diagnosis of this bacteria. Stool culture by using a rectal swab, and the rectal swab or stool can be cultured on McConkey agar for detection of non-lactose fermenter property, or the oxycolate agar and XLD media. Isolinate growth also can be used for inhibition the growth of normal flora and enhance the growth of Salmonella and Shigella. Microscopy can be seen leukocyte and RBCs. Biochemical tests including triple sugar iron agar, where this bacteria no produce gas, no produce H2S, and give acid to alkaline. Non-motile serological tests also can be used. Hanging drop can be used for for detection of motility. Gram stain, this bacteria is a gram-negative bacteria. MVIC test give indole positive, methyl positive, while vocal cross-cover and citrate utilization is give a negative result. Anaerobic fermenters, slide agglutination test with specific hemagglutinin or antibody can be used for detection of this bacteria. Commonly used primary isolation media including McConkey agar, Hector-enteric agar, and Salmonella Shigella agar. This media contain bile salts to inhibit the growth of other gram-negative bacteria and pH indicators to differentiate lactose fermenter from non-lactose fermenters such as Shigella. This is a type of culture media that used for isolation of and detection of Shigella. For treatment of cases of shigellosis, mild illness by rehydration, short signs and symptoms and duration, about 48 to 72 hours. Shigella soni, bacillary dysentery, using antimicrobial chemotherapy, shorten the duration and prevents spread. Ampicillin for plasmid resistance is present and prevent the make the bacteria is resistant to ampicillin. Cotrimoxazole, ciprofloxacin, and cibitriaxone also can be used for treatment of this bacteria. شباب اكو سؤال لحد الان؟ شباب واضح ان شاء الله؟ Now, prevention by using sterilization of water and use of good water supply, personal hygiene, sewage disposal, food hygiene, insect control, especially fleas, and using oral vaccine that give immunity for 6 months. شباب اكو سؤال بخصوص المحاضرة؟ شباب منو عنده سؤال؟
دكتور.
واضح دكتور إن شاء الله.
واضح ان شاء الله. ماشي. شباب بالنسبة لي قضية الدرجات مالتكم، انتم مطالبين بتقرير اللي يوصل الاسبوع الجاي ان شاء الله.